Yes, I'm Allan Dickerman. How do I move to the next slide? Kayaking the fire hose of information. And I, like Ina, am in the biosystems division and don't do lab work myself. I just analyze data. And I want to just give you a flavor of some of the sort of case studies or just examples, fun examples of data that I've been working on in the last year that were that I've found interesting, and some of it might spark some interest in some of you. On the both of these slides or figures represent a kind of molecular clock analysis that is a computationally intensive. There's a Markov chain Monte Carlo. It's a resampling of the likelihoods of different trees that can fit the phylogenetically organized data. On the left, we have porcine epidemic diarrheal virus. That's a serious challenge to the swine rearing agriculture, and it's swept into North America in about the last 6 years. And we have collaborators in XJ Meng's lab who is interested in the analysis of this virus in the American pig industry, and well, it's pretty obvious that it came from Asia because that's where it was rampant shortly before it came here, and they wanted to put a date on the time of arrival of PEDV into North America, and they had sequenced a strain of the virus, and there were 2 other existing sequences of the virus from isolates in North America. And there were several complete genomes of the virus from samples spanning a number of years in Asia. And this small blue square represents the isolates in North America, and you can see that they've got a very short phylogenetic distance among them. And knowing the dates from several of the isolates in Asia, we could put an estimate of, I think the estimate was for the spike, the peak of the blue, the probability distribution of the time of the divergence of the American strains was about 3 years. Now that's the divergence among the ones that are in America. So the arrival into America was the ancestor of those. So that is a lower bound on the time of arrival, and the upper bound on arrival was that next age group, which is about 7 years. That next peak has a bound of around 7 years. So between 7 and 3 years is the time of arrival of this virus into the pig population in North America. Then I'm working on a similar analysis in mosquito vectors of malaria, the Anopheles genus. And there's an interesting story on the divergence of New and Old World Anopheles, and I'm trying to see what the correlation is with the biogeographic, the separation of the continents and the opening of the Atlantic Ocean. I suspect that we can't pin the separation of New and Old World Anopheles on the opening of the Atlantic Ocean. It's subsequent to that, but it's interesting. Another whole area that I really like to work on is gut microbial diversity analysis, and people sometimes call this metagenomics, microbial metagenomics. I think real microbial metagenomics should be limited to when you're sequencing genomic DNA from around the whole genome. These kinds of studies typically focus on amplifying small subunit ribosomal RNA, so you're not analyzing the whole genome, you're analyzing a marker, but a very informative marker that you can put into a taxonomic context with some level of precision. And these particular data are from a horse gut microbiome in a study where they were using a probiotic to see what shifts in the microbial community you observe. And in this particular plot, I mashed together a few different things. You can plot the isolated strains or samples along with, and you can't read them, but some of those labels are from full genomes pulled out of GenBank. So that's and put them on a phylogenetic tree. I think that's as close to nailing down the relationships of your sampled small subunit ribosomal RNA sequences to, quote, known genomes. That's as fine a resolution as you can get, but it's too fine, so it's not suitable for summary back to publish because you've got, say, 500,000 different branches on your tree, and you can't visualize a tree that way. So you usually end up putting it into pie charts and stuff like that. Over on the right, this, quote, heat map is showing, well, the two axes on it are samples and not taxa, but biochemical functions enriched or present in the taxa. So that was an attempt to go from samples and changes over time to biochemical potential of the microbiomes involved. Just kind of running through these because I'm sampling a bunch of different kinds of studies. This is another, I have to say that the basis of all of this that I do is the capability of doing very, very dense, very high throughput, but high throughput isn't really the word, but large volume data production. And this is showing microRNAs mapped onto the genome. And just in general, analyzing microRNAs is another great field. It's really interesting, and you can always find new things. And in this case, I'm showing a novel microRNA in the mouse genome that we're working on. And finally, this is a new project that I'm doing with my wife, Rebecca Wadham, and she's got a collaborator at St. Jude's Hospital, St. Jude's Children's Hospital, and this is analyzing presence or absence of different strains of Staphylococcus in this case. Staphylococcus, presence or absence of genes in those genomes associated with disease phenotypes in the patients that they occur in. And we're trying to do a statistical analysis of the association of genes in bacteria with phenotypic outcome in the patients. And we really need Ina's insight on that. Next.